Hi everyone, welcome to Physiology for Students. In this lecture, we will talk about cancer cell biology. We're just finishing up our first unit, where we've talked a lot about the introduction to cell physiology, cell structure, the cell cycle, inheritance, and the basics of genetics. We're going to finish up looking at cancer cell biology. Most of this is not in the Guyton textbook, but it is a very important concept, and I think a lot of you will find it really interesting. So we're going to talk about common terminology in cancer cell biology. We're going to discuss the process of cancer cell transformation, and we will look at the characteristics of cancer cells. This is going to help us review some of the basic cell biology and the genetics that we've covered so far. Cancer is a term that encompasses many different diseases. The definition of cancer from the National Cancer Institute in the United States is diseases in which cells divide without control and are able to invade other tissues. Cancer is among the leading cause of death worldwide. In 2018, the most recent statistics that I have here, there were 18.1 million new cases and 9.5 million cancer-related deaths in the world. So despite a lot of work and dedication to the treatment of cancer, cancer is still a major problem worldwide. One thing I want to say before we get into the science is that cancer is personal. Most of us have family, close friends, or members of our community who've been affected by cancer in some way. In our family, we work with this incredible organization, Campo Kaizu. Campo Kaizu is a place where children with cancer and their families are supported every summer through an incredible camp program. Campo Kaizu was recently affected by devastating wildfires. If you'd like to learn more about Campo Kaizu and support their mission, please go to okaizu.org. So, on to some common cancer terminology. It's important to understand the basic terminology so that you can read pathology reports and understand the basics of benign and malignant tumors. A tumor simply means a swelling, but it has now primarily become used to refer to an abnormal growth. There are benign tumors and cancerous tumors or malignant tumors. A benign tumor is non-invasive and non-cancerous, but sometimes because of its growth can still be harmful. A malignant tumor is a cancerous tumor. And I'm going to show you the difference between benign and malignant tumors next. You may also hear the term neoplasm. Neoplasm means new growth. So neoplasm is often also used to describe a tumor. If you look in the McCant's pathophysiology text, you'll see a comparison between benign and malignant tumors. So just because someone has a tumor doesn't mean they have cancer. It means they have a growth. And then the tissue will be sent to pathology to decide whether it is a cancerous or a benign tumor. Benign tumors are generally slow-growing, encapsulated, and well-differentiated, meaning that they have a capsule around them and they're not spreading to lymph nodes or to distant sites. They're also rarely mitotic, meaning that they're not growing rapidly. Remember that mitosis is the process of cell division during cell growth and cell reproduction. Malignant is a cancerous tumor or a malignancy. Malignant tumors often have a more rapid growth rate. They have visible mitotic cells as they're growing rapidly, and they have some other important features of malignancy. There's a term called anaplastic. Anaplastic means they lose differentiation, or they no longer look like the normal mature cell type from which they came. Another term is pleomorphic, and that means morphology or their shape is irregular. So irregular size and shape of the nucleus in particular is characteristic of malignancy. Often, malignancy has a loss of normal tissue structure, and it can begin to lose a capsule, meaning that it no longer has defined borders and it has a chance of spreading. So, cancer or malignancies are invasive. In fact, the word cancer means crab because some of the early pathology of tumors showed these spreading like projections that made them crab-like in their appearance. Metastasis is another important word that refers to when cancer spreads to distant sites. Now, not all malignancies have reached the point of metastasis, but once they do, then they are becoming more and more concerning. This is an image from the McCants textbook, and what I like about it is that side by side it shows benign versus malignant features. On the right side you see a benign or leiomyoma. On the left side you see a malignant or leiomyosarcoma. So take a moment here to pause and look at the difference between the cartoon of the benign and the malignant tumor and see if you can pick out some of those characteristics. In particular, notice that the benign tumor is slower growing, it's more encapsulated, and it's not spreading or invading to the nearby tissue. Whereas the malignant tumor is getting larger, it's growing faster, and it's starting to become very irregular, spread and invade into the nearby tissues. If you zoom in and you look at the cellular features, you might see some of those other cellular features we described for the malignancy, meaning the anaplastic morphology, looking less differentiated, the pleomorphic cells and nuclei, meaning irregular shape and size. You're going to hear a lot of tumor names, and we could spend an hour just on looking at different tumor names. Of course, pathologists are going to help you with your tissue and tumor identification, and this is a very specific course of study for defining and deciding what type of tumor it is. But I want to give you just some of the basics here. So tumor names are variable, but there are some select differences in the naming of malignant versus benign tumors. Often tumors are named for the tissue of origin and whether it's benign or malignant. So for example, a carcinoma is an epithelial-derived tumor versus a papilloma, which is a benign tumor. Adenocarcinoma is a ductal or glandular-derived tumor versus an adenoma, which is a benign version in the same tissue. Sarcoma is a connective tissue-derived tumor versus other benign tumors from similar connective tissues might be a fibroma or a lipoma, chondromas, osteomas, etc. Lymphoma is a lymphatic tissue-derived tumor, derived from the lymph nodes and related tissues. Leukemia is a white blood cell-derived tumor. Leiomyosarcoma is what we saw in the previous slide. There are also historical names for tumors, which you will learn as you learn about various different cancers. So let's talk a little bit about cancer cell features and characteristics. So adult cancer is a disease of cumulative genetic or epigenetic changes, meaning there's multiple changes that happen to create the differences from a normal cell to a malignant cell. Transformation is the process by which a normal cell becomes a cancerous cell. In oncology terms, transformation is the process in which a cell changes to become cancerous. Let's look at some of these features here. I'm going to zoom in because there's a lot of words on this slide. So transformation from a normal cell to a cancer cell, you will often see these features of the newly forming cancer cells. First of all, they have autonomy or loss of normal cell cycle control and the decreased need for external growth factors. So normally growth factors would be limiting the growth of these cells. Now they're no longer being limited by growth factors for some reason. Sometimes that's because they're producing their own growth factors or because the cell signaling pathways have changed. They also often lose contact inhibition, meaning when they touch other cells, they're no longer listening to the fact that they should stop growing when they bump into other cells. And so they have continued growth despite a lack of space. Cancer cells often are anchorage independent, meaning that they don't need attachment sites the way that normal cells do, where they're not listening to the attachment sites. They often are immortal. They continue dividing despite the number of cell cycles. Anaplastic cells are cells without form, and they appear abnormal, as I showed you previously, as a hallmark of malignancy. Pleomorphic are cells with many forms or variable size and shape, particularly of the nucleus. And undifferentiated cells no longer look mature, and they're not fully specialized as the cell type that they should be. So when a pathologist gets a tissue sample, they're going to be looking for these features of cancer cells. How do cells become cancerous? One theory is clonal selection, and that is as cells in the body transform from benign to malignant, they may outcompete neighboring cells, meaning that they are actually growing more efficiently and faster than the neighboring cells. So they have less inhibited growth and less dependence on nearby cell signals. This creates multiple progressive and stepwise changes in the cells as they begin to get more and more aggressive, and they can have more and more changes because with every cell division cycle, you have the opportunity for more division and more mutation. This pushes changes then in favor of malignancy. Genetic changes to a cell lead to cell transformation to cancer. These could be direct genetic mutations or also epigenetic alterations. Remember that genetic mutations are changes in the DNA that can alter the expression or function of a cell. Epigenetic effects are alteration of DNA by molecular additions that can turn on or off certain genes, promoting cancer cell formation. One of the ways in which these genetic mutations can happen is through exposure to mutagens in the environment. So mutagens are agents that cause genetic mutations and increase the frequency of DNA defects. They increase the likelihood of cancer cell transformation. Normal body cells that are exposed to mutagens, somatic cells, can become cancerous. In that case, that will affect the individual with the exposure and can have cancer cell transformation within those body cells. That can be a particular cancer, but it won't be inherited. If the mutagens, however, are affecting the reproductive cells, then that can actually cause not only cancer cell formation, but cell changes that can be inherited by offspring. So the probability of mutations increases with exposure to certain mutagens. For example, ionizing radiation, carcinogenic chemicals, and physical irritants. These all increase the rate of mutation. Epigenetic changes are chemical modifications of the DNA that can silence or alter the expression of genes. This can be methylation, acetylation of histones, chromatin, or DNA. This can be faster than a full mutation of DNA sequences. There's evidence that epigenetic changes can, in fact, be inherited. And current research shows that epigenetic changes in many forms of cancer. There's also a link to cancer and many viruses. So viral infections are linked to many cancers. Some well-known viral infections that lead to cancer are human papillomavirus, or HPV. It's the cause of 80% of cervical cancers and also increases the risk of anogenital and penile cancers. The Epstein-Barr virus, which causes an increased risk of B-cell lymphomas, Burkitt lymphomas, and nasopharyngeal carcinomas. Kaposi sarcomas, herpes virus. Kaposi sarcoma, especially in immunocompromised individuals. And then hepatitis B and C. These chronic infections can lead to a twofold increase in liver cancers. There are many others and still others yet to be discovered. Here's an example of HPV, or human papillomavirus, causing cancer. The images here are of cancer of the cervix. So what you're seeing is a transient infection in a normal cervix to an HPV infected cervix, and then a chronic infection that becomes persistent HPV, precancerous, and then leads to cancer. What you see is that with prolonged HPV infection, you have viral DNA integrating into the cellular DNA and increased production of viral oncogenes. Remember that what the virus wants to do is increase proliferation of the virus. And so as a result, some of these viruses increase proliferation of cells. And remember, with increased proliferation of cells, you have the risk of increasing genetic mutations, increasing inflammation, and cancer cell transformation. Hanahan and Weinberg classically described the hallmarks of cancer. The early descriptions of the hallmarks of cancer, they named 6, and now we have an updated 10 hallmarks of cancer. This is an image from the McCants Pathophysiology text. It's also from the Robinson-Cochran Pathologic Disease text. These 10 hallmarks of cancer we're going to go through one by one in the next few slides. So this began as a study in 2000, updated in 2011. The initial hallmarks of cancer describe genetic alterations, and that is sustained proliferative signaling, evading growth suppressors, no longer listening to the suppressors of growth, genomic instability, increasing the likelihood of mutations, and enabling replication immortality, so increased cell cycle without those stop signals that are so important for preventing the cell from continuing to divide. The next group of the hallmarks of cancer are the cellular adaptations that allow tumor growth and cancer cell growth to continue. They have increased blood supply, or increased angiogenesis, and they have some interesting differences in metabolism that allow them to have an advantage against the other cells nearby. Finally, cancer cells evade cell destruction, and they do that by resisting apoptosis, promoting inflammation, the kind of inflammation that evades immune destruction and increases proliferation. And then finally, all of this leads to the activation of invasion and ultimately metastasis. So let's start with sustained proliferative signaling. In the cell cycle lecture, we talked about the ways in which growth and transcription factors regulate the cell cycle. So let's look at that here. So remember that growth and transcription factors are molecules that regulate the normal entrance into proliferative or growth phases of the cell cycle. If they are decreased or inactivated, that reduces proliferation, and that keeps the cells in check. Remember that many mature cells don't proliferate unless they need to be replaced. There are certain types of genes that are characteristically regulating the cell cycle that can lead to cancer. So oncogenes, when activated, can increase cell proliferation. Proto-oncogenes are genes that regulate these proliferation pathways. When proto-oncogenes are turned on, they become oncogenes, and the cell cycle continues unchecked to allow proliferation of these cells. Another hallmark of cancer is exactly the opposite. Instead of turning on cell proliferation or turning on oncogenes, they stop suppression of the cell cycle. So they evade growth suppression. In other words, they have an inability to stop the cell cycle. So anti-oncogenes are also known as tumor suppressor genes. These genes normally stop the cell cycle. If they're mutated, the stop signals are lost. And just as in the oncogenes, proliferation goes on unchecked. So compare them. Oncogenes are turned on to cause cancer. They increase proliferation. Tumor suppressor genes are turned off to increase cancer because the stop signals are no longer present. Inactivation of tumor suppressors then can cause cancer. A couple lectures ago, we talked about the example of P53, a classic tumor suppressor gene that normally halts the cell cycle to allow DNA repair and related mechanisms to happen. If P53 is mutated, individuals are at a very high risk of certain forms of cancer because the cell cycle can go on unchecked. Another genetic change that can cause one of these hallmarks of cancer is immortality. So enabling replication immortality can create cancer cells with an unlimited lifespan. Cancer cells have many more cycles of cell division than normal cells. Normal cells are normally limited by the ends of the chromosomes, and that is the telomeres. They shorten with each cycle of cell division. The telomeres shorten and shorten and shorten until eventually those cells can no longer replicate. Germ cells, stem cells, have an enzyme called telomerase, and in cancer cells, this enzyme is also often present, creating cells that no longer have shortened telomeres after each cycle of cell division. And this makes them essentially immortal. Their lifespan is not limited by the number of cell divisions, so they can continue dividing on and on and on without shortening their telomeres. Replacing the telomeres then allows that continuous cell division, and these cells can be immortal. Okay, some of the cellular changes that allow tumor growth to continue. Tumors are supported by a lot of blood supply. So tumors actually increase angiogenesis, or blood vessel growth nearby. Tumors larger than 1 millimeter require an additional blood supply to continue. So they will actually secrete angiogenic factors, or molecules that increase blood vessel growth to that tumor site. Another cellular change that increases tumor growth is reprogrammed metabolism. So cancer cells are often modified to perform more glycolysis than normal. So rewind a little bit to metabolism. Remember that glycolysis is an anaerobic process that allows a small amount of ATP formation in the absence of oxygen. It turns out that many cancer cells prefer glycolysis even when oxygen is present. This gives them the ability to create ATP no matter what environment they're in. Classically, this is called the Warburg effect, and that is glycolysis happening even in an oxygenated environment. Remember, though, that glycolysis doesn't produce a lot of ATP. So in order to make enough ATP, cancer cells have to do a lot of glycolysis. And what that means is that they're going to be using a lot of glucose, and they have some cellular and metabolic changes. For example, changes in the GLUT1 transporter to have high glucose utilization. So cancer cells will be using glycolysis and the lactic acid production that comes from that to make ATP. So they have a different way of metabolizing than the normal cells around them. Finally, the group of cancer hallmarks that are related to evading destruction or the inability to be destroyed. Cancer cells are resistant to apoptosis or cell death pathways. They suppress apoptosis and are resistant to cell death. Remember that apoptosis is programmed cell death, normally used during tissue remodeling or protection against abnormal or aberrant growth. So normally an abnormal growth should turn on the apoptotic pathways, but cancer cells often suppress those apoptotic pathways so they're not being destroyed. So the genetic and epigenetic changes that we talked about previously may lead to alterations in the apoptotic pathways. In other words, the normal programmed cell death is being prevented, and these cells can continue to live despite the fact that they are abnormal and growing abnormally. Inflammation is another very interesting area of cancer research. Cancer cells promote inflammation, and cancer is also linked to chronic inflammatory processes. So tumor-promoting inflammation shows that chronic inflammatory diseases, chronic viral infections, chronic bacterial or parasitic infections can predispose to cancer. Also, chronic inflammation can recruit similar inflammatory cell types and promote continued proliferation. Classically, within the stroma or the area around the tumor, there are a lot of inflammatory cells. And it's not typically the inflammatory cells that are there to destroy the cancer. It's actually often the group of inflammatory cells that the tumor cells themselves have recruited to hide from the immune system. So inflammation and the role of inflammatory cells in cancer, I imagine, is going to change a lot in our research in the next few years. Here's the most recent chart from the textbook about inflammatory conditions associated with neoplasms or tumors. I'm listing them here for you so you can see just how many inflammatory conditions are related to cancer, and I imagine there will be many more as the research continues. So as I said, cancer cells have an interesting relationship with the immune system. Not only are they caused by chronic inflammation and they promote inflammation, they also evade immune destruction. Normally, inflammatory and abnormal cells should recruit cells of the immune system and be destroyed. A normal, healthy immune system should be able to destroy cancer cells. Normally, T cells and natural killer cells will recognize and kill cancerous or abnormally proliferating cells. They have certain cell signaling mechanisms that will be recognized by the immune system, but somehow they're evading the immune system. They have tumor-associated antigens that can flag them for the immune system, and these antigens will be presented on MHC1 proteins, flagging them for destruction. But there's suppression of tumor antigen expression in MHC1 receptors, or there may be inhibition of T cells, and that allows persistence of cancer cells. Finally, the last hallmark of cancer is invasion and metastasis. As I said earlier, these are later stages of malignancy, but they're very important hallmarks of malignancy. Invasion is when cancer cells migrate and move locally into surrounding tissues. They release attachment to and degrade the extracellular matrix nearby. They're avoiding apoptosis, and they're also becoming mobile, moving into areas that they shouldn't be. Metastasis is when the cancer cells spread from the origin to tissues, to distant tissue sites. They can do this by entering the bloodstream or the lymphatics and traveling to sites throughout the body. They can spread to distant tissues and then survive and proliferate at new sites. One thing to note is that different forms and locations of cancer have different patterns of metastasis. And as you learn more about the different types of cancer, you will also learn about those that are more likely to spread to certain sites. For example, some cancers are more likely to spread to the brain or to the lungs, etc. Okay, I know this was a very brief overview. I hope it was interesting. I hope it was helpful. If you'd like further reading, you can find a little bit in Chapter 3, but much more in the McCants Pathophysiology text in Chapter 12. Feel free to add any comments below if you have additional questions, and I do have one challenge question coming up for you if you'd like to test yourself.